Good afternoon, humans. I'm going to go over the structure of DNA and RNA. This is my second video, so hopefully it's slightly better. I tested the audio. I sound terrible, so I do apologize. But I did pre-record my drawings, so hopefully this goes a little bit faster and easier. So we're looking at the DNA structure, and typically it's drawn in this like cartoon format, but it's not really what it looks like. It's just a simple way to draw it to help us better understand what's actually happening in the DNA. The DNA has two sides. It looks kind of like a twisted ladder. It's typically called a double helix. Double because it has two sides, and helix is that shape, kind of like a spiraling staircase. And that's why it's called the double helix. It's actually made of this molecule or monomer called nucleotides, which have three parts: a phosphate, a sugar, specifically a deoxyribose sugar, and then it will have one of four bases. The four bases that are in a DNA molecule are adenine, guanine, cytosine, and thymine. Adenine and guanine are called purines, and they're called purines because they have a double ring shape. Those two circles, the hexagon and pentagon. And cytosine and thymine are pyrimidines. They have one single ring, one single hexagon shape. There are some base pairing rules, and basically in the base pairing rules, purines and pyrimidines are going to bind together. A is always going to pair with T, adenine and thymine, and you can remember that as apples and trees. C is always going to pair with G. You can remember that as cars and garages. Cytosine and guanine are going to pair up. They kind of fit and match up together. This is just drawing out what these nucleotides actually look like. So we've been looking at this double helix shape, but in reality, it's these nucleotides that fit together, and they're going to bond together. These bases bond together through a hydrogen bond, and it's a pretty weak bond. I kind of think of it as magnets. It's helpful. They stick together, but they can easily be pulled apart, and we're actually going to see that next week when we go into details about protein synthesis. So this is actually what it's showing, the fact that we're drawing this cartoon, but in reality, it's these nucleotides that are bonding together. Here we're looking at, so we identified between the bases are hydrogen bonds, but on the outside of the bases are these sugar and phosphates. And these sugar phosphates are held together by a stronger bond called a covalent bond. These covalent bonds are really strong, and the backbone of the DNA molecule is held together by these covalent bonds. And they don't come apart very easily. You would need a really strong enzyme to break those down in that DNA backbone. And you would require a lot more energy to break it apart. So this is just looking at this DNA structure again, identifying in this cartoon rendering that these outside, the outside of the ladder are going to be the sugar phosphate backbone. And then the steps of the ladder are going to be the DNA bases. They're going to be on the actual steps of the ladder. And this is just saying A pairs with T, G pairs with C. So for looking at the bases along this ladder, one side, I gave you the strand for one side, A, G, G, C, T, T, A, C. Then we can fill in the complementary bases. And the complementary bases are going to be what they pair together. A goes with T, G goes with C. So if I give you this sequence, A, G, G, C, T, T, A, C, you should be able to identify T, C, C, G, A, T, G. A goes with T, G goes with C, and vice versa. T goes with A, and C goes with G. So this is said to be complementary. So if you see that language complementary, it just means that they fit together. they go together. And the complementary sequence is the sequence that goes with an original sequence. Well, because we know that A pairs with T, we also know and can figure out how much of each one goes with the other one. So if A pairs with T, A equals T. So if you're given that 20% of the DNA is guanine, you could figure out the percentage of thymine. As soon as I paused it to figure out why my screen got really dark, it stopped being dark. So if 28% of the DNA is G, we need to figure out the percent of the DNA that is going to be T. Well, since G pairs with C, whenever there's a G, there's a C. Whenever there's a C, there's a G. So the number of G's is going to equal the number of C's.
Joan, I need you to come to the main office.
So 28% of the DNA is G. So now we know 28% of the DNA is also C. We add that together, we know that 56% of the DNA is G and C. You subtract that from 100, we get that there's 44% of the DNA left. And since there's 44% left, we know that's A and T. Divide that by 2, and we know that T is going to be 22% of the DNA. And we know that just from knowing one base, you could find the percent of all the other bases in the DNA. So let's move on to RNA structure. RNA structure, obviously, is similar to DNA. They're both nucleic acids, but it's a little different. It's a little more unique. It's much smaller than DNA. We're going to see that its purpose is much different. DNA's purpose is to store and be like the backup information for the cell, for the organism, while RNA is more of a temporary molecule that takes a small bit of a gene. We're going to see that later on. It's going to take a small bit of the gene and it's going to transcribe it into RNA, and that will leave the nucleus. It's really small, so it can leave the nucleus to actually build a protein. So it's single-stranded, so it only has one side. It's not going to have that double helix. It's going to have that one side to it. Its sugar is going to be ribose instead of deoxyribose. It's going to have similar bases, adenine, guanine, and cytosine, but thymine is now replaced with uracil. You are the difference in RNA. You are the difference in RNA. U is going to be different. Uracil, also a pyrimidine, very similar to thymine in structure. It's just slightly different, so it has a different name. So base pairing, very similar to DNA. Not sure why the camera's dipping out again. G is still going to go to C, and A is still A is now going to pair with U. Before we remembered apples and trees and cars and garages. Well, our cars are still in garages, but now our apples are under. Apples under. A is going to pair with U. So just think about the U as replacing the T when you're on an RNA molecule. So if you're looking at the nucleotide, DNA is made of nucleotides. RNA also made of nucleotides, sugar, phosphate, and a base. But now that sugar is going to be a ribose sugar, and some of the bases are going to be U instead of T. The other thing about RNA is that they're unpaired bases. So they still have steps, but it's only going to be one base. We're going to see later on why it's going to be one base, and it is going to pair up to things, but by itself, the RNA is not going to pair up with anything. Okay. Let's keep going. There's 3 different types of RNA. They're going to play a major role in protein synthesis, each in a different way. mRNA is the main RNA we'll be looking at. It's called messenger RNA. It's going to carry the DNA message from the nucleus to outside the nucleus, the cytoplasm. Specifically, it's going to bring it to the ribosome. Ribosome, tell me more. The ribosome is actually made partly from rRNA, which is ribosomal RNA. It's not just rRNA. There's also other associated proteins in this structure. But when you hear rRNA, it's going to be ribosomal RNA, and it partly makes up the ribosome. The ribosome is actually what's going to translate the DNA message from the mRNA into proteins. It's going to be read the code to figure out how to assemble the proteins. Our last type of RNA is going to be tRNA, transfer RNA. Its job is going to be to transfer. It's also convenient that it's T-shaped, the tRNA T-shaped. Not quite, but a little bit. It's going to be transferring the amino acids to build up the protein to the ribosome as protein synthesis is happening. So it's going to play a major role in building up the protein on the ribosome by reading the mRNA code. Hopefully this is helpful. Please leave comments if there is something I could do to make it a little bit easier to read and listen to, and also subscribe to the BioMeek channel. I've been telling my students I could become YouTube famous. So please, I can only do that if you subscribe.